Hello, my name is Professor Matthew Schmidt, and welcome to Genetics. In this session, we are going to focus on the cell division process of meiosis and its relationship with the sexual life cycle. If you've watched the meiosis video, if you've watched the mitosis video, as I suspect you have, we want to start off by highlighting the similarities and differences between mitosis and meiosis. You may recall mitosis, the whole job of it is to make two identical daughter cells from one parent cell. We also said that the whole point of meiosis was in fact to be making cells that would become gametes or sexual cells. So meiosis is intrinsically linked with the sexual life cycle. Now, I don't want to really go into this in a lengthy fashion, but being human, we think of sex in a certain way, and really a sexual life cycle in a more purely biological sense is a much broader idea. For example, there are animals that do have a completely fine sexual life cycle from a biological point of view, where the male and the female don't even ever see each other, or probably they don't even know who each other are. Many fish would fall into this category. Plants are also sexually reproducing in many cases, and clearly they do not have activities related to the ones that animals do. The flower is the sexual organ of a plant. Pollen from one flower often will fertilize an egg from another flower. So the bottom line, what I'm trying to say is that the sexual life cycle is a very definite and fundamental biological process. It may be physically accomplished in a variety of ways. We may be interested in that, but we're not interested in it right now. So if we look at the board, we want to see that all, and I mean all, whether it's us, a plant, fungi, whoever is sexual reproducing, and that includes a great amount of the organisms on this world. All sexual life cycles contain two processes, and those two processes are complementary, meaning you sort of can't have one without the other. So meiosis, which we're going to be focusing on today, the whole point of it is we know it's to make gametes, but it's to reduce the chromosome number from 2n to n. And we spoke of that in the last video. So, but it's got to be clarified here why this has to occur. Because if a male makes sperm, for example, and a woman makes eggs, if they were humans and they had 46 chromosomes each, and if they came together, now you'd have 92 chromosomes. And if you follow that logic, every generation the chromosome number would increase. That would not be feasible. So what we've got to do is we've got to get the chromosome number down in half, and then fertilization is that process that puts it back together again. So meiosis you can think of as a reductionary process. It gets the chromosome number from 2n down to the haploid value. Fertilization takes an egg and a sperm or their equivalent and unites them, I should say. But what I mean is puts those things back together so that n plus n here algebraically gets us back to 2n. Then we've got a zygote, which is going to become a new organism one way or another. Now, how does meiosis accomplish this? During meiosis, it turns out that two complete division cycles occur, meiosis 1 and meiosis 2, aptly named, and each division has the same stages as those in mitosis. So there's, in other words, prophase 1, metaphase 1, anaphase 1, telophase 1. Then there's prophase 2, metaphase 2, anaphase 2, and telophase 2. Now, a lot of the things that go on in mitosis, there are similarities with the corresponding phases in meiosis, but there also are some key differences. So we're going to try to highlight the differences in this session, not recapitulate everything that we just discussed about mitosis. So remember, meiosis only occurs in sex cells, cells that are going to give rise to gametes. These are often referred to as the germ or germline cells. In fact, germline is really the opposite of the term we introduced last time, which was somatic. Somatic cells and germline cells together make up all of the cells. The germline are those cells that will make more cells that will create future generations. With that understanding, let's go ahead and really look back on a concept we touched on last time, but now we really want to make sure that we've got it because this is vital for understanding meiosis. Here we have a pair of homologous chromosomes, all right? The blue ones came from the father, they're paternal. The red one, well, I should say the red one came from the mother because we're seeing here not only a diagram but a process. Remember, before S phase, you have a homologous pair. You've got a chromosome with one chromatid, that's all it is, and the blue one is similar to that. So what I want to highlight here is that this would be true for mitosis or meiosis, that when they say duplication, we're talking about S phase here. So as S phase occurs, the chromosome gets doubled, and now here we have, just like we saw before, we said it looks like an X, and literally the two chromatids, remember now this is one chromosome composed of two chromatids, they're identical, so they're sister chromatids. The same thing has gone on over here, so these two are also sister chromatids. Now, the relationship of homologous pairs is an interesting one. They're not identical. I mean, the sister chromatids on each one are identical to each other, but the relationship of this to this, as you see here, those are called non-sister chromatids. They're not identical, but they do share something in common. And to illustrate this, go back to the idea of a locus that was introduced a while back. Say that the gene for eye color was on this chromosome. Say this was chromosome number 1. What I mean is this. So let's just say eye color. This person had a big B allele at that location. The other allele for eye color would be located the exact same spot on the other chromosome. So in that sense, they have in common the same genetic map, the same loci. And in fact, since these are identical to each other, we should fill in that's the way that it would be. So while the blue and the red homologous chromosomes are not identical, they have in common that they have information about the same traits. Now, I said this was true of mitosis or meiosis, which is correct, but the reason I'm highlighting it here in the meiosis talk is for the following reason. I hope it was sufficiently clear, but if not, let me just say, during mitosis, the homologous chromosomes really took no notice of each other whatsoever. We're about to see that during meiosis, they take great notice of each other. This diagram is just to make absolutely sure that you get the relationship between different types of chromosomes. During the course of this lecture, for technical reasons, the color scheme changes a little bit sometimes, so I'll try and keep you up to date with that so there's no confusion. For example, in this diagram, this is a homologous pair, and they've made them roughly the same color but different shades. This diagram is okay that way because it's just meant to illustrate a different point. And these are two homologous chromosomes. So the two that are directly attached are sister chromatids, right? We said that these two would be non-sister chromatids. This is a homologous pair. This is a different homologous pair, right? So they show them all of the things I'm mentioning are shown somewhere on this diagram. So they're showing that this and this, or any of those two from the red and the sort of pinkish looking one, would be non-sister chromatids. Also, if you were to compare this large one with that smaller one, those are just non-homologous chromosomes right there. If this is chromosome number 1, maybe that one's chromosome number 5 or whatever, right? So we have just getting the idea that there's one homologous pair, there's another one. They really don't have anything to do with one another, but the terminology stays constant with respect to who's a sister chromatid and who's a non-sister chromatid. Now, as we get into meiosis, here's an overview of meiosis. And I do apologize, the colors here are not going to be the same as they're going to be in a moment. But this is just to illustrate the point, and then we're going to see how it's accomplished. So if you look at this starting cell, what they've drawn is, again, they've drawn the homologous chromosomes in the same color. But here you'll see we're just trying to get down what happens differently in meiosis than mitosis that allows us to reach our goal of creating 4 haploid gametes. Okay, so this starting cell up here, it basically similar to the same starting cell you would have genetically similar during mitosis. 2n equals 4. It's before S phase, you can tell that. And this is one homologous pair, this is another homologous pair. Chromosomes duplicate or double during S phase. So after S phase, you see that it's the same idea, but everything's just been duplicated, right? So what you need to take away from this is the point of meiosis 1. After meiosis 1, look what's happened. We've separated homologous chromosomes. One of them went into one cell, one of them went into the other. And with respect to the other pair, they were separated. So what we end up with, you guys, very, very important. This cell right here, it is haploid. But it's odd looking because the chromosomes are still doubled. How do you know that it's haploid? Well, count them. There's only 2 chromosomes there when we started off with 4. They're still doubled because during meiosis 2 mechanically is much more like mitosis in that the sister chromatids are going to be pulled apart from each other. So after meiosis 2, you can see one sister chromatid here went to one cell, one went to the other, and we're ending up with 4 haploid gametes, just like we said, but we need 2 division cycles to allow this to occur. But be very clear. Division cycle 1, meiosis 1, the whole point of that is to take one whole doubled chromosome and separate it from its homologous partner. And then the point of meiosis 2 is to separate the sister chromatids. So while the ideas are similar, they're not identical. I think this will be borne out if we follow the process a little bit more closely. And now we're back to this is similar coloration as we did when we examined mitosis in detail, okay? So really this cell, even though it's drawn a tiny bit differently orientation-wise, this is a very similar, it's the same as the one we used to illustrate mitosis. What I mean is we're back to looking at it as though the red chromosomes came from the mother, the blue ones came from the father, right? This is a homologous pair, and this is a homologous pair. So we're going to assume, I'll mention it, but I'm not going to write it all down. You know, what happens in prophase happens in prophase both in mitosis and meiosis in the sense that the nuclear membrane's breaking down, the centrosomes are moving, the chromosomes are fully condensed, right? That's part of prophase. But in meiosis 1, prophase 1 of meiosis 1, look what happens. Homologs begin to associate with one another. So if you compare this with the mitosis diagram, in mitosis, the homologs randomly, they didn't have any interest in the other homologous member of their pair. Here, right away, the homologs are coming up very close to each other and becoming joined. Okay? Now, this is when a process called crossing over occurs. We're not going to get into a great deal of detail about crossing over, but it would be really remiss not to mention what's going on now. So think of it as we're going to take a tiny side trip because metaphase 1 is the next phase that's going to happen in meiosis. But the whole point of a sexual life cycle, I guess I should have mentioned this before, is to generate genetic diversity. And crossing over is one of the main ways that this diversity is accomplished, or created, I should say. Because when the 2 members of a homologous pair are joining up during meiosis 1, during prophase of meiosis 1, they're literally physically touching one another. And these areas here where they're literally touching, each of those is called a chiasma. Comes from the Greek letter chi, which looks like an X. and the plural is chiasmata. But the point of what's going on here, they often call this whole structure a bivalent. It just implies that the two doubled chromosomes are coming together here. So look, yes, these are two sister chromatids. These are two sister chromatids. It's the non-sister chromatids that really are going to, what's going to end up happening is they're going to exchange segments. So we've drawn it here this way to show you there's a physical, literal connection going on here and going on here. And what's going to happen is, if you follow, say, this up from here, all of a sudden, at this point, the blue chromosome is going to connect to the red, and then at this point, it's going to connect back. So basically, this entire segment from here to here is going to be switched or flipped between those two chromosomes. If we look at it in a little, not more detail, but this can get a little bit messy. So if you look at this diagram, over here, crossing over is occurring, and between what we've labeled as chromatid number 1 coming up here and chromatid number 4 coming over here, there's an intermingling there, right? And basically, here and here are the two chiasmata. I just noticed that it says here chromatids 1 and 3, but really, well, it's hard to say. It looks like chromatids 1 and 4 to me, but really it doesn't matter all that much. In this drawing, it looks like, in the drawing over here, it looks like 1 and 4, but over here, it's 1 and 3 that have switched segments. This is a technical, I'm not even sure if it's an error. I may just be looking at it the wrong way. But the point is, if you disentangle all of this, you'll find that 2 of them, 2 and 4 over here, are untouched. But if you look at 1 and 3, this segment right here has literally been traded between those two chromatids. So now, if you look at the chromatids, they were supposed to be identical, those sister chromatids. They're not identical anymore. This is a way of mixing and matching information, and it's critical to the development of genetic diversity in the gametes, okay? Now, this is very important, but when we go back, as we are now, to discussing what happens in meiosis, we're not going to draw the chromosomes with all of these different switched parts of them because that could become confusing. So when we go back here, it's going to look like they're just either all red or all blue. But just bear in mind that this crossing over event has already occurred. Now in metaphase 2, the cell equator now is in a different orientation, but it really doesn't matter because the cell doesn't know what's up or down or left or right. The most important part of this is the homologs remain together and they line up as pairs on the cell equator. The bottom line, guys, if you understood the summary is, we're going to separate this whole one from this whole one, this whole one from its partner, this whole one. And basically that's the point of meiosis 1, remember, is to get a whole doubled homolog away from its partner. In anaphase, it still looks like a V, but it looks like a doubled V, right? So this entire doubled chromosome is being pulled toward one side. This one's going to the other side. And basically, now guys, you could see, just like we did with mitosis, if you cut that right there, you're going to have 2 new cells. But look, they are going to be haploid because 1, 2, there's 2 chromosomes there and there's 2 there. They're not genetically identical at this point, and that's the whole point of meiosis, right? I should mention, so let's just be clear. The homologs are separated. Spindle fibers are pulling them apart. I'm not giving all the details like we did with mitosis because I don't think it's necessary. But just to go back here for a minute, we have to draw it one way, but I want you to be clear. It happened to be that a red one was on this side and a blue one was on this side, right? This is the whole basis, you guys, of independent assortment. The fact is, we really want to make that connection with Mendel, with independent assortment. Why is it that the segregation of one pair of alleles had nothing to do with the segregation of another pair of alleles? It turns out the reason was because Mendel's traits were on different chromosomes. So let's just say, what if flower color was on this chromosome and we had a big P, little p, right? And I don't know, tallness and shortness were on this chromosome. When these move, the alleles associated with them move with them. My point is this. So we drew it so that the red and the blue were on one side at the top and vice versa on the bottom. But it doesn't have to be that way because they have nothing to do with each other. So when you got to this point, it could have been that two blue ones ended up in the same cell. Here we got a red and a blue. It's completely random. And that, I'm not sure if it's going to be clear why, but that explains independent assortment. And that's so important for making the connection between the chromosomes movement and the behavior of what Mendel saw. Well, now I think you're going to get it because telophase, of course, is, the end part here. So what we've gotten here are two, this is haploid, but they're still duplicated. And of course, if we cut there and there's a cytokinesis event, that's not technically part of meiosis for the same reasons we said before, but it still has to happen. Now we're going to launch right into meiosis 2. And what's going to happen there, so during late interphase, what happens is it's like a resting period. So in other words, it's as if you're going around, I don't want to obviously make it confusing. You don't go through the full cell cycle twice. So in this situation, just think of it as a resting stage before meiosis 2 begins. Whether it should technically be called interphase or not, I might have to think about a little bit. But we've got what we just said, two haploid cells, each of which has doubled chromosomes. In this case, there happens to be a blue with a red in each cell, but it doesn't have to be that way. And then we're going to go into prophase 2. So again, all the things you would expect, the nuclear membrane breaking down, the chromosomes condensing, the centrosomes moving. We're keeping track of each cell here now. You can already see they're not genetically identical, yet they are both haploid. And what's going to happen is, in metaphase 2, again, this is the cell equator, and it's like mitosis in the sense that we're going to pull apart these sister chromatids in each of these cells. When we go into anaphase, you can see them being pulled apart. And you can also see that once we make that cut, we're going to end up having 1, 2, 3, 4 haploid cells, which are not all genetically identical. Now, these two look identical and these two look identical, but that's not taking account of crossing over because that has happened and it's caused a lot of mixing and matching. Okay, so all four of the gametes are not going to be genetically identical. When we get into telophase, as you might suspect, this is the end phase. The nuclear membranes are reforming, etc., etc. But the bottom line is we now have 4 haploid cells where n equals 2, right? And each of them has a chromosome with only one chromatid. So guys, you need to understand that really the difference between mitosis and meiosis is the way that original material, which is it's the same in both cases. It's the way in which it's partitioned out that allows you to either get 2 genetically identical diploid cells or 4 haploid cells that are not genetically identical. All right? Now, the whole point of these is to make gametes, and gametes come in a lot of different flavors depending on the organism. But let's say this was a human and that they're going to be made into sperm and eggs. The term gametogenesis refers to the making of gametes. Meiosis is always the genetic process that leads to it, but as we're going to see, men and women make gametes differently. The daughter cells here, there are 4 of them, right? And we're back to each single chromatid now being referred to as a chromosome, or you could say that the chromosomes are comprised of one chromatid. Let's look at the male version of gametogenesis, which is called spermatogenesis, perhaps not surprisingly. And actually, males are very straightforward in this respect, in that all of the products of meiosis become functional sperm. So we're not going to go through the whole process again, and unfortunately the colors are back to the other way that they were before. But the idea here is This original cell that's diploid, we're seeing it after S phase, right? That is called a primary spermatocyte. After meiosis 1 is complete, we have two haploid cells, right? And those are referred to as secondary spermatocytes. Now, each of those undergoes meiosis 2, and you end up with four so-called spermatids. Genetically, that's done. They're only called spermatids because they're not mature yet, and they need to go in a structure called the epididymis, where they're exposed to various hormones in order to develop into actual functional sperm cells that can swim, and they're differentiated. Now, if you look really, really closely, you'll see that there's little chromosomes in the nucleus of these sperm cells. For the reasons we've discussed, all four of those sperm are going to be genetically different from one another, which I hope it's not becoming too redundant, but which is the whole point of meiosis. Now, the process of meiosis works the same for females, but as you're going to see, there is a major difference in the timing of events and the result. And the reason makes sense because human females only wish to release one egg, one mature egg at a time. There are millions and millions of sperm present potentially, and if 4 eggs were released by a female, likely 4 eggs would be fertilized, and that's not the way human body is built. So in females, only one functional egg will result. 2 other cells will be pushed off to the side, and we're going to call them polar bodies. And instead of spermatogenesis, we have oogenesis, which is a little bit embarrassing to say, oogenesis, but an egg technically is called an ovum. And in fact, the word egg is used a little bit loosely by many, many people. And sometimes it's no big deal, but as long as we're being technical, think of the same process going on, so we don't have to discuss all that. The original cell, instead of we had a primary spermatocyte for men, we have a primary oocyte for women. I don't know if you've heard this, but it's true. It's often said that girls are born already with all of the eggs that they're ever going to have in their lifetime. And in a very real sense, that's true. But more technically, what they're born with are all of the primary oocytes that they're ever going to have. And the process of meiosis 1 begins very early in utero, but it arrests. It stops right in the, not quite the middle, but it stops. And it remains arrested until puberty. All right? In fact, the only time meiosis 1 is going to occur here, and they call it an asymmetrical division for reasons you'll see in a moment, is that this only occurs, by the way, at ovulation. So when an egg matures from its follicle and it's released, that's the signal to complete meiosis 1. Once it's completed, you get one functional secondary oocyte, which is haploid. Now, genetically, this other cell it's not identical, but it's haploid, and it could be viable. But it becomes a polar body. It's deprived of cytoplasm, and it's sort of pushed off to the side because this is the only one that's going to be doing any work. It's the only one that's going to become an actual, egg is perhaps not the correct term. The reason why is because this primary oocyte often gets called an egg. This secondary oocyte often gets called an egg as well, right? Because once ovulation occurs, you say egg, but really this is what's going on. And meiosis 2 only occurs, you guys, if a sperm comes over and actually fertilizes this. If fertilization doesn't occur, meiosis 2 does not complete. It starts again, but it arrests until fertilization occurs. So technically, an actual mature ovum only exists for about a second, if that, because as soon as that sperm comes in, meiosis 2 finishes. Another polar body gets thrown off, and you end up with this mature ovum, which immediately joins with the sperm to form a zygote. So the timing, it makes sense in a way. You could say, if it's not going to get fertilized, why bother? It's just going to go out, right? So But regardless of the explanation, this is what occurs. So you might expect that the total number of haploid cells created were 4, but look at the difference between these two polar bodies. One of them is still doubled, so most of the time that polar body, it doesn't bother dividing again. It could, but what's the point? I mean, whether you ended up with 2 polar bodies or 3 polar bodies, none of them have any function anyway. So again, in summary, between men and women, the general process, the genetic logic of meiosis is really the same, but the timing and the end result in terms of actual gametes are different. Meiosis is a topic that is confusing to some, so I strongly suggest if you have to watch this more than once, obviously that's your option, and I encourage it if you want. Also, check out the problem-solving questions that we have. Often through challenging yourself with problems, you'll gain an even greater conceptual understanding. Well, that concludes our discussion of meiosis. I hope you enjoyed it as much as I enjoyed discussing it with you, and I hope to see you soon in another genetics session.